Hello students, today we are going to look at the details of fertilization. We have already started the topic yesterday, and in yesterday's class we had discussed the structure of pollen grain, what is the detailed structure of the ovary, that also we have seen. And as I told you earlier, the plant reproduction or the sexual reproduction in plants involves two steps, the pollination and fertilization. Pollination has been done already, means the pollen grain is landed on the stigma, okay, and the next step is fertilization. And the first process in the fertilization, or see, fertilization means the fusion of male and female gamete, okay. It is not that much a simple process, or it is not a single step process. In the case of animals, the male and the female gametes are fusing directly, but in the case of plants there are 2-3 steps involved in between, okay. So firstly, the pollen grain is landed on the stigma, and first step is that the identification of the pollen grain, means the interaction will be there in between the pollen grain and the stigma. The pollen stigma interaction, that is the first step. The stigma is identifying its own pollen grain, means the genetic features should be similar to the stigma and to the pollen grain, otherwise the pollen grain is not germinating, okay. And after the identification, it will start to produce one liquid, a sugary liquid the stigma will produce, and that is actually starting the germination of the pollen grain, okay. And the sugary liquid will get absorbed by the pollen grain, and that's the first step of the germination of the pollen grain. Yesterday I have told you that you can see that there is a portion where there is no exine. Exine means the outer covering of the pollen grain. There is a portion without exine, and that is known as a germ pore. Through the germ pore, the pollen tube is arising, okay. And you have to recall that the pollen grain is having two cells. One generative cell is there, and one big tube cell is also there within this pollen grain. And after the secretion of the stigma, it will absorb this one. And what is really happening means the generative cell inside the pollen grain will get divide, okay. The generative cell within the pollen grain is dividing, and it is undergoing one mitosis. Actually one mitotic division it is undergoing, or the generative cell is undergoing one mitosis, so that we are getting two similar cells or two male gametes that we are getting, okay. So the pollen grain, within the pollen grain two cells are there, one tubular cell is there and another one is one generative cell. And when the stigma is producing the sugary liquid, it will get absorbed by the pollen grain and will start the process of germination, okay. And the generative cell is undergoing one mitosis, and afterwards we are getting two male gametes, okay. And what is the next step? The pollen germination, otherwise the development of the pollen tube. The pollen tube will get developed. Pollen tube will get developed. So how the pollen tube is developing? Through this portion is known as for germ pore. Through the germ pore, the pollen tube will develop, okay. It is developing, okay. And what are the contents inside the pollen grain? All these things will be entered into the pollen tube. What are the contents over here? And one tubular nucleus will be there, tube nucleus will be there, that we have seen already, and two generative nucleus, otherwise two male gametes will be there, okay. Male gametes, okay, and the entire cytoplasm. So everything inside the pollen grain is going to enter into the pollen tube, okay. So I am just erasing this particular things from here, and I am drawing here, okay. The tubular nucleus will be there, and two male gametes and the cytoplasm. Everything will get entered into the pollen tube, okay. So I am just drawing this here, a big picture that I am drawing here. See, the stigma, this is a stigma and this is a style. The pollen tube is passing through the style, and sometimes the style will be hollow and sometimes it will be solid. What do you mean by solid? The inside the style, so many cells will be there. Okay, so many cells will be there inside the style, and it can be hollow also. See here, it is hollow. Here in this style, this one is hollow, no cells are there, but here it is packed with so many cells. So when a pollen tube is arising, what is really happening means, when a pollen tube is arising, this pollen tube is secreting some enzymes. Okay, the pollen tube will secrete some enzymes, and that enzymes will dissolve these cells within. Okay, and the cells are getting dissolved so that the pollen tube can easily pass through the style. Have you understood? So the pollen grain is started its germination and it will form the pollen tube. Pollen grain is forming the pollen tube. Sometimes the style is packed with cells, means it is not a hollow one. Sometimes it will be a solid style, means it is packed with so many cells. And how the pollen tube can pass through? The pollen tube is producing some enzymes. By means of these enzymes, the cell which is packed inside the style will get dissolved so that it can easily pass through the style. Okay. And finally, it will reach up to the micropyle. Micropyle means this portion where there is no covering for the ovule or there is no integument. Okay. And when it is reaching here, the further process I will make here in this diagram. This is, as you know, this is what? This is the embryo sac. Embryo sac is within the ovary. So what is really happening? See, the pollen tube is coming up to this. Okay, the pollen tube is coming up to this, and when it is reaching up to this, the tubular nucleus will get dissociate. Okay, there is no need of tubular nucleus then, and the tubular nucleus will dissociate, and now it is having only two male gametes. Okay, two male gametes will be there. And you know what is the name of this particular cells in the either side of the egg? The center one is egg, and these two are known as what? Synergids. Synergids. Okay. And the synergid is having a special structure over here, and that is known as what? Filiform apparatus. Okay, it is known as filiform apparatus. So the filiform apparatus of the synergid is giving the way to the pollen tube to enter in. Okay. Otherwise, simply you can say that the synergid is giving the way to the pollen tube to get into this embryo sac. Okay. So the pollen tube will enter into the synergid. One of the synergid, not both. One of the synergid will take this pollen tube inside. Okay. And after, when it is entering into the synergid will dissociate or it will disintegrate, and the pollen tube will break. Okay. The pollen tube will break, and when the pollen tube is breaking, what will happen? The male gametes will get released in the embryo sac. Okay. So the pollen tube is coming up to this, and two synergids are over here. One of the synergid will help the pollen tube to enter into the embryo sac. Okay. When it is getting entered into one of the synergid will dissociate and the pollen tube will get break. Okay. Due to the breakage of the pollen tube, the male gametes will get released in the embryo sac. Now the male gametes are there in the embryo sac, and one of the male gamete will fuse with the egg. Okay, one of the male gamete will fuse with the egg. Okay, one of the male gamete is fused with the egg, and it will form the zygote. Okay, it will form the zygote. Okay, so first fusion has happened, and the second nucleus will move to the center. You know that in embryo sac there are two polar nuclei are there. One of the male gamete will move to the center towards the polar nuclei, and it will fuse with the polar nucleus. Now it became a cell which is having three nucleus. Okay, three nucleated structure, because it has two nucleus over here and one more male nucleus is fused with that, so that it became a 3-nucleated structure, and it is forming the primary. This is forming the primary endosperm. That is forming the primary endosperm. So two times the fusion has been done. One time the male gamete is fused with a centrally placed egg to form the zygote, and one of the male nucleus is fusing with the polar nuclei, and two more nucleus were there. One more nucleus is going to add with that. So the fusion of 3 nucleus has been done. So that this fusion, the fusion of the male gamete with the polar nuclei, we can call it as triple fusion. What we can call it as triple fusion. Triple fusion means what? Triple fusion means here already 2 nuclei were there. One of the male nuclei is going to add with the centrally placed polar nuclei, so that 3 nucleus will be there. So it is a triploid cell, means it has got 3 end nucleus, so that we are calling it as triple fusion. Okay. So 2 times of fusion has been done here, so that the entire process that we can call as what? Double fertilization. Double fertilization. So why it is a double fertilization? Because 2 male gametes or male nucleus were there. One of the nucleus fused with the egg to form the zygote. One of the nucleus fused with the centrally placed polar nuclei. So that 2 times of fusion has been done, so that it is known as double fertilization. So you can get the questions, what is triple fusion, what is double fertilization, okay, and what will be the fate of the egg, what will be the fate of the polar nucleus. So these kind of questions that you can get from this portion. Okay. So what are the next steps, or what are the post fertilization changes that we are going to look in the next class. Thank you.